right, so we're going to go from very big to very small, I think, because we're moving into molecular type work. And I thank everyone for allowing me to come and talk to you about biomarkers. This is what I do in my lab, and this is what I love. And hopefully all of you will learn to love it too. I don't get a dime from this, unfortunately. I think it actually costs me a ton of money. But our lab is funded through the EDRN, and I probably do have agreements with everything I'm going to talk about in terms that we do share specimens with them all. So for a biomarker to be useful in cancer, it has to do a couple of things. It really does have to reduce the burden of the cancer. It has to be able to alter treatments and outcomes, and that seems obvious, but it's not always so easy. And it also has to do more than just detect GGOs. You know, it can't just detect the indolent cancers. We have to be able to get the more aggressive ones. When we talk about cancer biomarkers, they do fall into 4 categories, and those are all very different. So we have early detection biomarkers, and those really tell us about the risk of developing a cancer. And I think of like the BRCA mutations as these early detection biomarkers. There's diagnostic biomarkers. So once we have something we think is a cancer, you know, is it a cancer or is it not? Then there's also prognostic biomarkers. They tell us about which patients, once they have the disease, have an aggressive version of the disease, and then predictive. And these are very much, you know, we hear about a lot now, predictive biomarkers, now that we have targeted agents that don't treat every single cancer. What does this mean in lung cancer? Obviously, early detection ones are the ones that we're going to use to try and, you know, determine who needs to go to screening and to direct that. The diagnostic biomarkers we're going to use to determine when we find a nodule, is that nodule cancer or not. Prognostic markers could be used to direct adjuvant therapy for our resected stage 1s, an area where there's tremendous need still. And then again, we've talked about predictive as being the ones that can help us choose the appropriate treatment now that we have this incredibly wide variety. So in early detection, where does this fit with CT screening? So I think all early detection and diagnostic biomarkers now have to work within the realm of CT screening. It's here and it's going to stay. Well, the early detection ones should be the ones that enrich that pool. There should be more than just age and smoking to tell us who needs a CT scan. Even good models like the Tamamagi really don't take any biologic things into account. The diagnostic ones should then take those CT nodules and tell us what it is. Again, we're determined mostly upon age, smoking status, and the size to tell us what do we do with a 6 or 8 millimeter nodule. Why is there nothing that within the biology of the patient or the biology of the tumor that can direct us better? Why should thoracic surgeons care? Well, one, this is our disease. Early stage lung cancer is us, and therefore we need to be there. We are the gatekeepers for these patients still, and we're also very much the front line in being able to collect not only tissue, but blood, demographics, and clinical follow-up. The reality is none of our large databases can do this. This is something that we have to be an essential part of. And when I think of where this comes in, one of our cohorts that is the most wanted by all people who work with these is we call them the abortive thoracotomies. Those patients who had enough bad prognostic things that we went in to biopsy them, and they were granuloma. That's the one that every company wants. Everyone wants that person, that group, because that's where we can really make differences. And we're the only ones who have those patients, because the pulmonologists already said they had cancer. They moved on. I have to say a word about the EDRN, because that's who does a lot of our funding. The Early Detection Research Network is a collaborative group funded through the NIH. It's funded in a very similar mechanism to the cooperative groups. Similar to the cooperative groups, it's had a major decrease in its funding as of late. But these are individual investigators, and the real power of this is that it's a network. So similar to what we talked about on Thursday, as a way for people to just come and talk, as much as the grants are important, what's important about the EDRN is that it really pushes collaboration, and it's a time twice a year for all of us who do this kind of work to get together and share ideas. It is important that we all get bonus grants for if you actually collaborate with other people. And what the EDRN is, it uses a really important structure to try and push biomarkers through development, and that includes hooking discovery labs with validation labs, and then with clinical validation labs. And these are all important. And the NIH defines these steps for biomarkers, and they go 1 through 5. And the EDRN will get you through the 3 phases. That'll get you to the FDA. But after that come 2 really important ones, which most biomarkers we have now have not gotten through phase 4 and 5. And this is determining the clinical benefit that a biomarker can add. And this is prospective evaluation and adding it to screening initiatives. And this is where we go from hundreds and hundreds at the start of the pipeline. And we really haven't gotten any that have made it all the way through from where we are. I'm focusing really on the early detection and diagnostic biomarkers because I think that's where we as surgeons have our greatest impact. And that's really working to enrich the at-risk population, to do better than the NSLT criteria for who we get to CT scans, and then trying really hard to reduce the number of invasive diagnostic procedures. When we look at platforms and sources, so the main sources for biomarker work, especially those that are pre-resection, pre-prognosis, would be things like airway epithelium, sputum, obviously circulating ones in the blood, and then exhaled breath has always been this interesting one that people really like. I wish I had Mark's slide from yesterday that would go through all of the genetic stuff that we can use for this, because any piece from the DNA through the protein to the tumors can be used. There are some obvious things which are missing here that you guys hear a lot about, not in the news, but in research. One is circulating tumor cells. We don't see that here. Another one is free DNA. Those are the liquid biopsies. For early diagnosis and detection, we're not seeing as much utility for those platforms. And that is, I think, really because the goal here is that we want to be working with tumors that are too small, hopefully, to shed their cells or their DNA into the serum. And a lot of these other things are the things that either are reactions by our bodies to what's going on with the tumor, or those things that put our bodies at risk for these tumors. So I'll start with Airway Genomics. This is, like, I always think the coolest of the things we've seen, and I think the one that has a lot of potential. The primary work from this is from Avi Spear at BU, and he works with really this field of injury effect, meaning that the RNA changes that one develops from smoking don't just happen in the tumor, they happen throughout. And this group is working to see whether an RNA gene expression profile in the large airway epithelium could help predict which nodules are tumors versus non-tumors in smokers. So again, the way your body or this individual's body responds to the tobacco would do that. And what he's identified is a profile of mutagenic changes in patients with nodules. And he has an 80 gene expression profile that can distinguish a cancer from a control with incredible accuracy, 80% sensitivity and 84% specificity. It's been taken through validation cohorts and really does a good job. This is not for mass screening. This is for the patient who has a small nodule and we don't have a diagnosis. Can this keep us from moving a certain patient onto resection or onto a biopsy? This paper came out just about a month ago from their group, and they wanted to see, is there added value to this profile in a group of patients who are undergoing bronchoscopies? So again, 188 patients with a nodule found on CT who undergo a bronchoscopy. They still don't have a diagnosis. They use the Tamamagi model to try and say, is this a cancer, is this a high-risk patient or a low-risk patient? And then they went on to add their genomic profile. And in doing so, they could have reduced unneeded procedures by about 50%. So again, taking these best predictive models we have based upon clinical phenotype demographics and such, and then adding a little bit of biology to it. Does this make a difference? And they say they could take about half the biopsies. That's, I think, an important step for our patients in terms of dollars and interventions. Even cooler, this is where it gets super cool. The field effect should go beyond the large airways. Could the field effect extend to your nose? And actually, they've determined that it could. So again, they looked at, this was just using, this wasn't really a nodule-based thing, but this was just using smokers and healthy controls. They were able to determine that the RNA expression changes that we see related to tobacco that happen in the large airways can also be mimicked in your nose. So again, something simple. It's not really inexpensive yet, but it could get there, that you could do with a CT scan to help you better risk stratify your patients for those guys who have nodules, who can be watched, and who needs intervention. Also, people are looking in the sputum. I put this slide up because I think you guys have heard a lot about microRNAs, those small non-coding RNAs, which for diagnosis and detection are really pretty good because they seem to respond faster than some of the genomic RNA you're getting out of the tumors. And they're easy to find. We find them in the blood and exosomes, and amazingly, this work from Maryland shows that they're incredibly stable also in the sputum. So again, none of these are perfect, but this has been validated that they can pick up miR21, which has been identified in multiple trials as something that's most likely related to cancer progression. It suppresses a tumor suppressor gene, and that it's stable in the sputum and correlates with the presence of cancer in patients with nodules. Biomarkers in the blood. I love blood biomarkers. They're easy to find, easy to get. Probably the king of microRNAs in the blood right now is this guy, Carl Croce from Ohio State. He's really done amazing work. This was a fairly important paper where he was able to determine actually many different kinds of things that blood-based microRNAs could find, defining risk, early detection, then diagnosis once people had nodules, and then even defining prognosis within this one study that used 2 large Italian screening trials. He set up all these profiles. All of these trials, also I didn't really mention earlier, they take these big group of patients and they set up 2. There has to be a discovery set and a validation set. And he did this with these 2 really nice trials, and these are 4 rock curves show you the first one is early detection, the second one is diagnosis, and the third is prognosis based upon these microRNAs. It's also important to note that when he looks at these microRNAs, that these patterns, these signatures are all different. They're not the same. And this is something that's sometimes difficult for people like me who's really concrete to fully grasp, that with almost all of these technologies, we're not looking for the BRCA1 gene. And part of that is because lung cancer is a really complicated disease genetically with so many different mutations, and therefore everything that we're going to be finding is this risk, and they're all going to be a little bit different. I don't think we're ever going to find a BRCA1 for lung cancer because our disease is just way too complicated for that. There's also epigenetics. Again, Mark discussed this a little bit yesterday. Again, these are not actual changes in your DNA, but things that control how your DNA is read and passed on. And this is nice work from David Sadransky at Johns Hopkins looking at patterns of methylation for tumor suppressor genes. And if you have one of these 4 and they're upregulated, you most likely have a higher risk of cancer. Again, this is for patients with nodules or with GGOs, and nicely it can separate them. What's the coolest about the epigenetics is that methylation is much more common in smokers. And this group could help determine cancers from non-cancers in non-smokers. So if you had increased evidence of methylation as a non-smoker, that was very, very predictive of having a cancer. Proteomics is another one. Again, some people think proteins are probably better than the DNA and RNA messages because it's the final product and so many things go into them. This is work from Pierre Massion, very smart guy from Vanderbilt. He found a 7-protein peak signature that was able to differentiate cancers and controls. And again, this is similar to the work from Avi Spira, added value. He went back and said, what does this add to what we already have? He looked at 2 cohorts, one from Vanderbilt and one from Mayo, and said, you know, what does this add to what we already have in terms of CT, age, and smoking? And surprising, it didn't add anything in their own cohort. But in the Mayo cohort, it was very helpful So again, that's made them go back and look. And again, in patients who were incredibly high risk already because they had 3-centimeter tumors and smoked, 100 pack years, this signature didn't really help. But in those that had smaller tumors and lighter smoking histories, adding this biologic difference, this little bit of biologic information really seemed to help in differentiating cancers from controls. Industry investigators, when the EDRN, we work with lots of this, has gone to companies. These are 3 super promising proteomic companies. One is SomaLogic, Celera, and then Integrated Diagnostics. All of these have gone through all 3 phases. They are very good. Each of them uses a slightly different technology with their proteins, but each has a signature that is very good at discriminating cancers from controls. And I'm sure some of you in the room have worked with these companies in the past because the list is very broad. Autoantibodies, this is another kind of interesting thing as we're all learning more and more about what the immune system means in cancer and even in very early cancer. While we can't necessarily pick up tumors in our blood, we can detect our body's response to them. This company, Oncommune, works on autoantibodies using just a basic ELISA type platform. It's been very successful. What's interesting about this, and the reason I included this, is that once they got FDA approval, instead of setting up a prospective trial, they actually just set up a prospective registry, and then every 6 months goes through to determine how well this is working within that. Part of the challenge of all of these is those, the randomized trial to prove benefit is long and expensive when you're doing when you're talking about early detection and diagnosis. So far, this technology has been good. A positive test, really a 5.4 increased risk of lung cancer. That would push most of us to go toward biopsy if we were looking at a 6-millimeter lesion with that. And then finally, breath testing. Again, people like this because it's kind of cool, it's kind of different. I think the original ideas for breath testing came from dogs, that dogs could detect cancer and no cancer in patients. So that's always cool, I think. And this is an early detection thing. This is an upstream thing, meaning that it can recognize changes in cytochrome P450 in our breath, which changes how we respond to the insults of tobacco. And that's how it works in being able to detect cancer from controls. It really can't smell the cancer. It can recognize that your P450 doesn't work appropriately. These are actually super easy to do. It sounds hard, but actually most of the things that work with breath tests, the patients breathe into them, you collect them, and you send them in regular mail. Ours doesn't even go overnight anymore. We just send them in literally the US mail back to the company who can read them. It's amazingly stable. And Mensana, again, has worked with many of the big institutions in the country. And again, it's not a one individual volatile compound that they recognize. It's a signature that is important because this one has as many compounds that go down as up. But again, very, very sensitive at recognizing cancers from controls, both in current and former smokers. Non-smokers are a real challenge for early detection because we don't have a lot of screening that uses them. It's also, again, because it's early detection, it's not changed by the stage of the disease. So in conclusion, I really think that biomarkers are going to be the key in how we work early detection and diagnosis. It's very obvious that CT scanning is a tremendous step forward for us, but we all know that it's incredibly expensive and that 96% of nodules detected on screening are not cancer. So we need something that improves upon that. Right now, there is a tremendous gap between promise and product. Obviously, none of these are anywhere near coming to the clinic, but I think some of that will get better as we move forward, and with screening initiatives, it has brought a whole group of new patients and new investigators into this pool who really weren't there before. Thank you.